Hello everyone, welcome to my channel Dr. Tapati's presentation. Myself Dr. Tapati Bhanjate. Today I am going to start a new topic of enzyme, that is enzyme inhibition. This is very important topic of enzyme. I hope it will be helpful for you.
I am going to discuss about enzyme inhibitor and enzyme inhibition, mechanism of action of inhibitor, importance of enzyme inhibition, and types of enzyme inhibitor. First of all, know what is enzyme inhibitor and what is enzyme inhibition. Various organic and inorganic substances alter the enzyme activity by binding with it in a way that influences the binding of substrate or enzyme's turnover number. Substance that decreases activity of an enzyme in this way is known as inhibitor. So we can define enzyme inhibitor as the substance that binds with enzyme and reduces the enzyme activity. Whereas enzyme inhibition can be defined as the process in which enzyme activity is reduced by any substance or chemical. As you know that in an enzyme catalyzed reaction, ES complex formation is the first step of this reaction, and second step is the ES complex breakdown to release product and free enzyme. Enzyme inhibitor may prevent formation of ES complex or may prevent ES breakdown to product and free enzyme. So it may interfere in the first step of the enzyme catalyzed reaction, or it may interfere in the second step. You can say that inhibitors work in two ways. They either stop the substrate from getting to enzyme's active site or prevent the enzyme from catalyzing the reaction. When inhibitor binds with free enzyme or ES complex, enzyme activity is reduced. So inhibitor either binds with free enzyme, so it will make EI complex, or inhibitor binds with ES complex to produce ESI. Let's know the importance of enzyme inhibition. Enzymes are required for almost all biological processes required for life. Enzymes catalyze a reaction by reducing the activation energy required for the reaction to occur. However, enzymes need to be tightly regulated to ensure that levels of the reaction product do not rise to undesirable levels. This is accomplished by enzyme inhibition. Metabolites regulate enzyme activity by allosteric regulation of substrate inhibition. Enzyme inhibition can provide insight into the mechanism of enzyme activity, as for example, by identifying residues critical for catalysis or identifying the active site of the enzyme. A lot of drugs act as inhibitors by blocking the activity of enzymes. Pathogens can be killed or metabolic imbalances can be corrected. Various plants and animals produce a variety of poisonous substances like peptides, proteins, and secondary metabolites that act as natural inhibitors. These natural inhibitors not only target enzymes but also target structural protein functions and receptor channels. They also used as neurotoxins for defense against predators or capturing prey. Inhibitors can also be used as pesticides. Acetylcholine ester is a crucial enzyme of animals for the nerve cell functioning. Carbamate pesticides are reversible acetylcholine esterase inhibitors, and malathion, parathion, and chloropyrifos and other are organophosphate pesticides. Those are irreversible inhibitor of acetylcholine esterase. Glyphosate, which is a herbicide, inhibits 3-phosphocyclimate 1-carboxyvinyl transferase, which is used to make branched chain amino acids in plants. So there are three types of enzyme inhibitor: reversible, irreversible, and allosteric. Reversible inhibitors are of three types: competitive, uncompetitive, and non-competitive. Irreversible inhibitors also categorized into three groups: group-specific reagents, reactive substrate analogues, and suicide inhibitors. Allosteric enzymes are classified into two classes, K class and V class.
Thank you very much for watching this video. Kindly like, share and subscribe this channel. Don't forget to press the bell icon for further notification.